I'm extremely pleased to be here, and I hope to entertain you for an hour. So I found, well, first about Max Birnstiel. I almost did my postdoc with him when he was still in Zurich, actually, 1983. That you can count back how old I was and how old I am now, and that's why I'm now emeritus. So, that's beside the point. But it was, so as fate will have it, I went to work with Uli Lemley and brought me to the dark matter of the genomic universe. Actually, I found the title of my lecture on the web because I had given something really boring, and then I saw that this was advertised, and I thought, that's a great title. I'm going to put it up. So I must have given it in some moment of inspiration and then fell back into my usual boring self. But the first half, I have two parts. One is completely on the dark matter, which is the silent parts, done by a postdoc and a graduate student using C. elegans. And the last part just got accepted in Nature this night. I got it while I was flying here, and it's done by Daphne. So it's about chromatin. Chromatin is the organization, the packing of our genome into around nucleosomes, and then the marking that the nucleosome marks in many cases lead to the status of the chromatin, the packaging, the folding, etc. This is going to be the first half of the talk, is all about repetitive elements and silencing. Most of the world cares about the open bits, but they're really a small fraction of the genome. And you can say it's the important part, but I hope to convince you otherwise. Just real fast, so we're on the same level. Polycomb is the classic facultative heterochromatin. That means it's cell type specific and cell type specific genes that can be in a repressed or a poised state. Sometimes polycomb it's certainly not irreversibly silenced, in that it can easily shift into an active state if the cell fate markers are there. Open chromatin, of course, you know, is unfolded, is active. Pol2 is able to bind, often transcribed, but actually there's an open state when it's not actively transcribed. And then there's the massive bit of constitutive heterochromatin, which is packaged away, usually not to be transcribed, but actually tissue-specific promoters in terminal differentiated cells also become canine methylated. And in mammals, the DNA becomes CpG methylated as well. A lot of our interest has been not just in the chromatin, but in the spatial, beyond that local packaging. You know, how is it packaged in the nucleus? And you know, the yeast telomeres were mentioned, but the fact that repressed chromatin is at the nuclear envelope is true through all species. And as nuclei, these are nuclei that are stained such that the yellow is heterochromatin, the blue is euchromatin. As you differentiate, internally differentiate, you accumulate more and more heterochromatin, often sequestered at the nuclear envelope or around the nucleolus. Every cell type will have a different pattern. Transformed cells often lose the segregation of heterochromatin, euchromatin, the clean organization. And what we do know about heterochromatin, and this is C. elegans nuclei, is that it accumulates repressive marks. So H3K9 methylations accumulated on the heterochromatic, dense staining chromatin, as well as, especially in worms, K27 trimethylation. I mean, these co-localize on many repressed domains, but not all. So our organism, as mentioned, of choice has been C. elegans, largely because it does differentiate, and there was limited information that we could gain by studying spatial organization in yeast. Not that it's not important, not that many of the underlying principles of chromosome structure were determined in yeast. But when we knocked out all the attachment of heterochromatin at the nuclear envelope in yeast, you know, the telomeres got a little bit longer. We de-repressed a lot of non-essential genes in subtelomeric regions, but basically the yeast cell didn't care. And that's not very rewarding in life to work on something where even a yeast cell doesn't care about it. So we launched when I moved to FMI about 15 years ago, and even before that, when I was in Geneva, I had been planning this for a number of years, that I was going to work on C. elegans. And it was easy in FMI because there was already a C. elegans group, and then we hired even more. So why work on C. elegans? I'm going to go really fast here. One is that for a yeast geneticist, it differentiates fast, and that is the crucial thing, that you don't have to wait weeks to get an answer. The second important thing is that the cell lineages are completely specified. You can determine a cell fate, know exactly when a cell fate is determined and what its lineage will be. There are not stem cell pools in these differentiated somatic cells, the 959 somatic cells of the adult worm. The stem cells basically are all in the gonad, the germline, but it's a fantastic system to study differentiation. And on top of that, it's transparent. And that has been crucial because I'm a microscopist at heart, not only. Here we're looking live at an embryo where we have a M-cherry lamin and a GFP tagging a tissue-specific gene. And you can do live imaging and very quantitative, as was mentioned, positioning on hundreds of nuclei that are, at least in the embryos, quasi-equivalent in their differentiation stage. So what we learned by tagging tissue-specific promoters and then some housekeeping promoters, only later, much later, looking at the heterochromatin itself, we learned that in undifferentiated embryonic nuclei, this for the C. elegans people, this was up to maybe 200 cell stage, between 100 and 200 cell stage. We didn't find any specific position, even for silent or active promoters. So we tagged, let's say, some muscle-specific genes, gut-specific genes, neuronal-specific genes, and they just didn't seem to care where they were. But the repetitive DNA, and when we put in a large array, which was repetitive, it was completely sequestered at the nuclear envelope. Now, then we followed these tagged promoters through differentiation. This was the work of Peter Meister. And we found that in every tissue where the promoter was active, the gene moved to the middle of the nucleus, and every tissue where the tissue-specific gene was silent, it was sequestered at the periphery. And this was, it was far cleaner than yeast. Yeast, everything was always 70-30, and this was really 90%, 95%, 95% in. Housekeeping promoters didn't care. So we set out, this is a perfect baseline to ask then the question, what does it matter? What sequesters? One, identify the mechanism through genetic screens, and then two, knock them out and ask, what's the effect on development? What's the effect on genomic integrity? So that's the menu for today. The first part is on the heterochromatin, the repetitive DNA part, so that not dealing with the tissue-specific genes. Why do we need to sequester repetitive DNA into heterochromatin, and what happens when we don't? Because our initial screens were to find out what was tethering the heterochromatin at the nuclear envelope. And the second part of my talk is what just got accepted. It's the forces at work at segregating euchromatin from heterochromatin in differentiated cells, in terminally differentiated cells. Now, why is that important? It's important because most disease hits terminally differentiated cells. You have diseases of liver, lung, organs. These are differentiated cells. Developmental diseases are usually lethal. So if we're interested at all in biomedical application of nuclear organization or chromatin spatial organization, it's really interesting to understand what leads to proper programs of gene expression, maintenance of nuclear organization. So first to the repeats. The reason I think repeats are important is because, in fact, they make up about 70% of the human genome. And there's something that no organism has actually managed to get rid of. Budding yeast did a pretty good job, but it still has telomeric repeats and still has a few transposable elements. And it does silence these, and that is our system of silencing. In mammals, about 50% of your genome, or 43, 45% of your genome, are made up of transposable elements. These are viral elements that have invaded your genome, and then we have, over evolutionary time, learned to live with them by silencing them. There are DNA transposons, LTR RNA transposons, long terminal repeat transposons, and then there are lines that have no long terminal repeats, and signs. All of them have Pol2 promoters, and if you look at the copy number, where if genes make up 1 to 2% of your genome, the vast majority of Pol2 promoters are in viral elements and not on your precious genes. So it's very, very important, actually, that we cope with this and that we have a way that these are not driving transcription or, let's say, creating a sink for transcription. Then there's all the tandem repeats. Telomeric regions have simple repeats, T2A3G. There are large amounts of the genome dedicated to the centromeric repeats, satellites in non-holocentric, so normal chromosomes. These range from 160 to up to about 200 base pairs of repeats. And then there are lots of microsatellites and mini-satellites. Micro are usually 2 to 5, and mini between 10 and 30 base pairs of nonsense simple repeat that's amplified in long stretches. So it's something that even pufferfish, this is one thing where Sidney Brenner was not 100% correct. He argued that pufferfish had gotten rid of, they even have about 12% repeats. And so there is no organism that has been able to eradicate these invaders. Mostly we've learned to live with them. Now in C. elegans, we have all the classes except centromeric satellites because they're holocentric, so they don't have the repeats around the centromeres. They have microsatellites, mini-satellites, telomeric repeats, and they have a larger fraction of DNA transposons than RNA transposons, but they still have all the classes. The interesting thing about C. elegans is that they're all mappable, so they're usually not in very, very large arrays, but in short arrays, so that you can actually tell if they're being transcribed or not, because you can usually find a primer pair or a sequencing pair that allows you to know where they are. That means we can map the chromatin state, and we can map the transcriptional state. So we did that, and others have too, but we've particularly focused on the repeats. The repeats are enriched on the chromosome arms, and of course at telomeres. The central core of all C. elegans chromosomes tends to be gene-rich. If we map the H3K9 dimethyl and trimethylation, you see that it's enriched on the chromosome arms, the extremities of the arms, and depleted from the core. These are the regions that are associated with the nuclear envelope, and the central regions usually loop in. But in fact, there are genes in these terminal repeat regions, and the genes, when they're active, they're actually pulled away from the periphery, and they'll be internal in the tissue where they're expressed. So the question was really, what's constitutive heterochromatin good for besides efficient centromere functions? And that was work done here by Antoine Peters and Thomas Genuine years ago, that when they knocked down SUVAR39, both homologs in mice, they found a lot of chromosome missegregation. In worms, there's no missegregation of chromosomes, not in meiosis nor in mitosis when you knock down H3K9 methylation. But we were able to study what happens when you get rid of all H3K9 methylation, because C. elegans has only 2 enzymes. In mammals, there are at least 8, and they're highly redundant, and it's not even clear always which ones are doing mono and di and tri. These are names I'm sure you know, the SETDB1 or ESET enzyme, the SUVAR39, the 2 I just mentioned, G9A and GLIP, which are also somehow related, and then these putative mono H3K9 methyltransferases. In any case, until recently, actually, Thomas has just managed to make a hextuple knockout in mammals, but when we were doing our work, nobody had been able to knock out more than 2 or at most 3 of these enzymes. So C. elegans, the MET2 enzyme deposits mono and di. I'll show you that. It's really clearly the SET-DB1 or ESET homolog. SET25 has a set domain that is very similar to SUVAR39 and G9A, and it is the only enzyme that puts on trimethyl. C. elegans has readers of H3K9, which are HP1-like factors 1 and 2, and of course has the modification. So how do we know what these do, and how can I state with confidence we can get rid of these? We made the double mutant and did mass spec on embryos and L1 larvae and normalized it to wild type levels. And you can see here, this is the unmodified K9. You get a little bit more when you have both of them knocked out. But mono, di, and tri are completely gone in the double mutant. If you knock out only SET25, then you have mono and di. So that means that MET2 is able to do mono and di, and SET25 the tri. So what were the phenotypes? So, you know, it was almost like yeast all over again. The bloody worms were fine. They grew, they differentiated. And I think that was sort of, you know, unexpected. But in the end, what it says is that heterochromatin is not essential for the programming of differentiation. And I just heard the same thing this morning. You know, it's really transcription factor driven. You push the button that says you're going to be, you know, this particular cell in the differentiated worm, and most of the programs proceed fairly well without heterochromatin determining which genes will be on and off. That's not to say that they're in perfect health, but they do differentiate. This is just staining to show that they really don't have any detectable K9 methylation. What we noticed right away by propagating the strain was that we were getting fewer and fewer worms as we cultivated them, and they are progressively sterile. This is actually only after 2 generations, but if you propagate them at 20 degrees, you see that they still are able to propagate. The yellow is the double mutant, the green is wild type. This is the size of the brood, the offspring pool. At 15 degrees, they propagate just fine. 20, it gets smaller and smaller with time. But at just 1 generation at 25 degrees, or 2 generations at 25, 1 at 26, you basically have no offspring. So there is a depletion or a germline lethality. We also looked at repetitive elements by transcription, and we found in the double mutant massive expression from tandem repeats. And as I said, we could map these because most of them are less than 400 base pairs long. So by long sequencing, we could map them. You see a massive derepression and derepression of transposons. Not every transposon and not every repeat, but many. About 37%, if you want a number. And then we noticed that they accumulated R loops and DNA mutations. So this was published in Nature Genetics a few years ago, and I'll just run through the data quickly. We singled the worms and sequenced single genomes to see what kinds of damage they were accumulating. I was thinking there would be lots of translocations and so forth, but actually what we found were insertions and deletions were the dominant mutation. So small insertions and deletions, sometimes just a few base pairs, sometimes larger. And then there were transpositions. And every time there was a transposition, there was a transposon. So obviously we were derepressing certain classes of transposons, and those that were able to translocate did. And then these insertions and deletions, there was no increase of single nucleotide variability. Now this would be germline mutations. And of course, we probably lost a lot of the mutations because they were lethal. So we asked the question, do you also get this in somatic cells? And so to do that, you can't actually isolate, or it's difficult to isolate single cells and sequence them. So what we did was use a LAC-Z gene that was inserted in the genome out of frame, and where you would have to have an insertion or a deletion to express LAC-Z. This worm is carrying it, but it's inserted as an array, and you see no LAC-Z. But if you mutagenize, or if in the case of the H3K9 deficient worms, we would find, and where it would be shifted back in frame, then we would see in which cells actually the frame shift occurred and what color it was. I mean, that it accumulated color. So what we found was that when the gene, the reporter, was on a heterochromatic array, the double mutant met set in 90% of the cells. They were at least 1/3 to a full blue, as defective as an MA6. So this is known for mismatch repair and actually also replication fork repair. Equal. So loss of H3K9 methylation was dramatically inducing small insertions and deletions. We sequenced them, and indeed, that's what we were seeing. Small insertions and deletions, putting that frame shift back in frame. So we asked the question, why do they arise? Were there double-strand breaks and misrepair or something like that? And the first hint came from exposing the worms to different kinds of stress, DNA damage. We thought maybe the double-strand break repair, or because there have been reports that K9 methylation associates with double-strand break repair. But gamma irradiation of wild-type and mutant worms showed the same survival curves. The first hint that there might be something to do with replication came from exposing the worms to HU. It's a transient exposure to HU and then asking them to grow afterwards. And we saw that actually the K9 deficient worms, that's the gray here, was very, very sensitive to even a transient exposure to HU. This is the wild-type survival. So that was one hint. There was something wrong with replication. The second hint came from doing a genome-wide screen. So this is, of course, what C. elegans is great for. We took the METset double mutant, which we know can propagate at 20 degrees, and we asked, what are the factors that are allowing it to survive? You know, we know at 25 you don't get any offspring. So we were trying to find out, so this indirect logic, that if you could find out what was compensating for the survival at 20 degrees, you would find what it's doing. At least that was the logic. And in fact, I think it's true. We screened at 20 degrees. Control would show plenty of growth, both in wild type, and so you plated 5 or 6 worms and looked 3 days later. You could see the offspring, these threads in the background. If you had a positive hit, we were looking for an absence of offspring, so an inability to propagate at 20 degrees. We had 109 hits. Not a single one of them was a histone methyltransferase. Of course, we thought, well, if you don't have K9, you're going to have, H4K20, or you're going to have K27 or some other modification. That's not the case. I'll show you the hits in a moment. But we then took all 109 hits and tested them in single mutants. And that was really revealing because all of them, every single hit, was synthetic lethal or synthetic sterile with MET2, and none of them was SET25. We did the screen in the double mutant, and remember, SET25 does the K9 trimethyl, and MET2 does the K9 di. And so what we learned was that these are the hits, the 109 hits. These are at least 10-fold reduced progeny, at least 5-fold, at least 2-fold. All of them were synthetic lethal with MET2, and none of them were SET25. Very importantly, MET2 deposits mono and di, and SET25, it will also do mono and di, but it's the unique trimethyl. So we tried to ask the question then, what is the difference between these two marks and these two enzymes? So we did transcriptomics on the single mutants, so MET2 versus SET25, and it was really clear. So here the brownish color is derepression. These are the simple repeats. These are certain sets of RNA transposons and other sets of DNA transposons. In SET25, we found both RNA and DNA transposons being derepressed. Obviously not all of them, but certain ones very strongly derepressed. MET2 had a partial effect on transposons, but not very strong. But MET2 uniquely derepressed simple repeats, all except maybe 1 or 2. And this was really unique to the MET2 enzyme. So it was H3K9 dimethylation deposited by the E set, set DB1, was repressing tandem repeats. And we checked by chip to see whether we lost H3K9 dimethylation. In RNA and DNA transposons, you got a partial effect because the two enzymes compensate. But tandem repeats, set25 had 100% of the tandem repeats maintained their methylation, dimethylation in this case, and lost it all in a MET2. So there was a clear separation of duties. MET2 is targeted to simple repeats, and this is one of its main jobs. So just remember that, ESET, MET2, it does do some DNA and RNA transposons, but can be compensated to a certain extent by set25. And set25 has some RNA and DNA transposons, which MET2 is not really needed for. But above all, MET2 silences tandem repeats. So we were still lost in this. Why did the cells care about tandem repeats? So we looked at the synthetic lethal. If you didn't have the silencing of tandem repeats, MET2, what was causing the death? Okay, what was causing the sterility? So these were the 109 hits of synthetic lethality at 20 degrees. And they came out in very clear classes. The bigger the symbol, the bigger the hit. RNA degradation and RNA processing were major hits. Some transcription factors, but interestingly, they were basal transcription factors, mostly. Some chromatin remodelers. The mitochondrial I'll skip, even though in honor of Jeff Schatz, I should certainly try to say something about it, but I really don't know why these were lethal. But the set of enzymes that interested it the most were a set of enzymes that are involved in replication fork stability. And we had already known that HU was important. So they're all old friends of mine, like TOPO2, CDC6, RAD51, ATR, that's the checkpoint kinase. I'd studied all of these in budding yeast. But budding yeast doesn't have BRCA1 and BARD1, and those came out as the strongest hits. BRCA1, BARD1, and ATR, and RAD51. There's SMC3 for those who care. I'm sure nobody here cares about SMC3, but you know, it was there. And BRCA1, BRCA2 popped up as interesting. Now, in the literature of replication fork stability, a large fraction of these factors had been identified as factors that are involved in R-loop resolution and replication fork stability. And specifically, BRCA1, although it's not the popular literature about BRCA1, everyone thinks BRCA1 binds at double-strand breaks, and that's what it does. In fact, more and more people are finding that BRCA1 is involved in replication fork stability. And there was a paper from Nick Proudfoot and David Livingston that had said that BRCA1 brings in an RNA-DNA helicase called senataxin that unwinds annealed RNA from highly transcribed genes in order to promote proper termination, that's one thing, but also to prevent damage in these highly transcribed genes. So BRCA1's associated or resolves R loops. BRCA1 together with RAD51 stabilizes stalled replication forks and prevents collapse. And it's associated with the transcription machinery at sites of replication transcription interference. This was all in the literature and made us very interested that 3 of our strongest hits were BRCA1, BARD1, and a cofactor that had, at least in the database, is it's an RNA binding factor that may interact with the C. elegans BRCA1, BARD1 complex. BARD1 is, of course, a H2A ubiquitin ligase. So there was this hint that BRCA1, BARD1, and a large fraction of our hits were associated with R loops. We had this HU dependency of replication fork instability with lethality, germline lethality. And so what is the link? Well, in the replication or fork stability field, replication fork progression is one of the most sensitive times of cell's life. And most damage and most actually chemotherapies that target cancer cells act in S phase to promote problems at the replication fork. So, of course, when the replication fork encounters DNA lesions, folded DNA, secondary structure, repetitive sequences, or, as we now know, RNA-DNA hybrids, the replication forks tend to stall. The longer it stalls, if it doesn't have a full complement of ATR or a checkpoint, the fork will become destabilized and create breaks or translocations. So this is one of the major roles now of what or problems associated with R loops. And so we ask the question, does H3K9, loss of H3K9, generate R loops? And in fact, it does. So we used an antibody. There's an antibody that specifically recognizes this. Here we're staining embryos. In the MET-set double mutant, almost every single nucleus had these bright spots of RNA-DNA hybrid formation, as much as TOC2 mutant, which is a known, it's involved in RNA processing and export and generates R loops. Interestingly, the R loop formation here in the double mutant was temperature dependent, so they were much higher at 25 degrees, at 15, it paralleled the sterility, and it was RNase H sensitive. We mapped where they were by something called RNA-seq, or DRIP-seq, DNA-RNA hybrid sequencing. So you use the antibody to pull down the DNA-RNA hybrid and then sequence either the RNA or the DNA. In fact, we did both. And in wild-type cells, you get very, very little specificity in what you pull down. What you do pull down, if anything, is tandem repeats. And in the MET-set double mutant, here you see a really dramatic enrichment for repetitive elements, not genes. Actually, exons are here. DNA transposons and tandem repeats. And just to remind you, that is the pattern of what's repressed by MET2. So what we concluded was loss probably of one of the HMTs. MET2 is the culprit. The synthetic lethality with BRCA1, BRCA2, and was true with the MET2 alone and not with SET25. And it seemed to be that simple repeat transcription was generating R loops, and it was far worse than transposon activation from SET25, which does occur. So we asked the question, can we document this genome instability? So first of all, the MET2 alone does have this brood size problem, SET25 doesn't. You accumulate RAD51 staining, which are signs of breaks or DNA damage or stalled replication forks. And when we combine MET2 with BRCA1, BARD1, we could reproduce this synthetic sterility that we saw in the original screen. Is the sterility linked to R loops and repeat transcription? So then we ask the question, does the MET2 alone generate the R loops, and does BRCA1 generate R loops? So indeed, the SET25, this is the background in wild type. SET25 alone, you do see some R loops, but the MET2 and the MET-set double mutant were almost equivalent. This is a quantitation. So you really accumulate high levels of RNA-DNA hybrids in the MET2 as you do in the double. And we asked, does that happen in BRCA1 null allele? And indeed, you do. It's a somewhat lower level, but we could detect the RNA-DNA hybrids by knocking out BRCA1 and actually by same thing with BARD1. We then did RNA-seq, or a DRIP-seq. So the RNA-DNA hybrid, pull it down and sequence it. In this case, we didn't sequence the whole genome because actually we had a limited amount of material, but we did qPCR. So we did DRIP qPCR, and we asked, where do we find the RNA-DNA hybrids? And we did qPCR for transposons, for unique genes, and for simple repeats. The yellow is the BRCA1, BARD1 mutant, and what you see is that all the RNA-DNA hybrids were enriched on tandem repeats, not on RNA-DNA transposons and not on simple genes. So transcription of repeats is really bad news. You become dependent on BRCA. You become sterile if you're a worm, and you generate a lot of replication fork damage. So we asked whether the BRCA1, BARD1 itself was de-repressing these repeats, which is not what you expect a repair factor to do. But indeed, this is RNA-seq, so we isolated total RNA from the BRCA1, BARD1, and this third factor, and just quantified a number of repeats, a number of transposons in a single copy gene. 1 here means no enrichment. There was a 2 to 3-fold enrichment of transcripts or RNAs from simple repeats just in the knockdown of BRCA1, BARD1. So it does do low-level silencing. But when we combined it with the MET2, we saw a dramatic synergy between the BRCA1 and the MET2. Now, if you're thinking, I'm BRCA1 minus, beware, because if they treat you with H3K9 inhibitors, your tandem repeats are going to be expressed. This is true in mammalian cells as well, by the way. So we found this, in the absence of MET2, you see a dramatic increase in transcripts from the BRCA1, BARD1, and these are the conditions, of course, that are synthetic sterile. The SET25, we could see that the transposons are de-repressed, but not the tandem repeats, and this is just the combination of the double mutant with the same mutant, with the BRCA1, BARD1, and this third factor. So the conclusion at this point is that BRCA1 complex is essential for the suppression of tandem repeat transcription. Maybe essential is a little bit strong. You do get a low-level transcription, but loss of BRCA1 is strongly additive with MET2 for all negative phenotypes. The BRCA2-dependent ubiquitin does mark MET2-dependent repressed tandem repeats. I didn't show you that. And in the absence of BRCA1, the H3K9 methyl drops. So the two complexes, the ubiquitin ligase and the K9 methyl HMT, do crosstalk. And in the long run, the important thing is that the two of them, BRCA1 and the H3K9 methyltransferase, is essential to prevent the accumulation of RNA-DNA hybrids, particularly on tandem repeats. So I've shown you lots of correlations, and what you come down to is you say, okay, you can correlate all these things, but is tandem repeat expression really the cause of sterility, the cause of R loops, the cause of the damage? So we did an experiment, a gain-of-function mutant, where we targeted a cleavage-dead Cas9 fused to VP160 so that we could turn on whatever sequence it was targeted to. And we targeted it to one of the major satellites, tandem repeats in worms that's present in about 220 copies. This just shows that we are able to make transcripts from these. This is in a completely wild-type background. And when we asked whether this was enough to cause sterility, indeed it was. Overexpression of tandem repeats, and this was an abundant tandem repeat, did cause sterility. And we asked whether it was, again, going through the DNA damage pathway, so we scored for apoptosis. Indeed, we could see high-level apoptosis, and again, it was, I didn't show this, but it is p53 dependent, and that's the argument that it's actually a DNA damage-induced apoptosis. And this is a completely wild-type background, and all we're doing is turning on one of the major satellites in the worm and creating this kind of germline sterility. So our take-home is that you don't need H3K9 methylation to differentiate. It's enriched on repetitive elements, and it's there that it may play its essential role, at least in evolution, to stabilize our genome. It excludes RNA pol 2. Now, if you lose H3K9 methylation, then for some reason these repetitive elements, probably through read-through or promiscuous initiation, they get transcribed. And the RNA DNA R loops accumulate, and then this becomes toxic when replication forks come through. So it's a combination, actually, of the H3K9 methylation on tandem repeats that keeps transcription away. If you lose this and you get transcription, then you're likely to generate DNA hybrids, and then these lead to the insertions, deletions, loss of germline integrity, genome instability, and there is a delay in development that I didn't show you. But this is not the whole story. We know BRCA1 is recruited in to recover from RNA-DNA hybrids by unwinding and degrading, allowing enzymes to come in to unwind these and relieve the stress. But we also find that the BRCA1 complex is contributing to the repression, and in part through the H2A ubiquitination. These both co-localize on tandem repeats. And what we would propose is that actually once you have damage, you've recruited BRCA1, it actually then deposits the H2A, and although we haven't proven it, we would suggest that maybe these two marks somehow cross-talk to reinforce the repression on tandem repeats. And this is how we survive having all this repetitive DNA. What happens when you have this? I said that the worms were viable without H3K9 methylation. Of course, they're sterile. They're also short-lived. If you lose both, of course, then your repeats become very unstable and your genome becomes unstable in general. And so I would say that these two marks may have evolved together in order to allow us to deal with all the repeats that have populated our genome. So once again, if you're BRCA minus, unless they're treating you with a tumor, I would be careful of G9A inhibitors. So that was the first part. Okay, in 10 minutes, I'm going to tell you our last story. That is the forces that work on segregating euchromatin from heterochromatin. This is going to have to be fast. The other thing about heterochromatin is that it's sequestered at the nuclear envelope. And when you get rid of MET2, you also get rid of this sequestration at the nuclear periphery. This is done by LEM2 chip, which is, and this is a worm chromosome, where you see the ends of the chromosome associated with the periphery and the middle away. And this just is wiped out by MET2. So the K9 methylation does more than just silent repeat. It also spatially organizes the genome. MET2 itself forms foci that are found at the nuclear periphery and co-localize with our heterochromatic reporter. So the enzyme stays associated with the locus. And we ask the question, are MET2 foci that you see here around the edge of the nucleus, are they dependent on readers like HP1, Lin61, or CEC4, which was the factor we found that sequesters heterochromatin at the periphery? The foci still form, even in a CEC4 mutant, they just shift inwards. So CEC4 is a reader of K9 dimethylation, which sequesters the heterochromatin at the periphery. HPL1 and HPL2 and Lin61 are all readers that are required for repression, but you see that the foci still form at the nuclear periphery. So what we knew was that there are factors. K9 does more than just silence repeat. It organizes the genome. Most of our work that was published was about sequestering heterochromatin at the nuclear envelope in embryos. But I remind you, embryos are a small part of an adult worm's life. It's the only period where cells are really undifferentiated. And then basically you're differentiated by the time you're in L1, and then you just grow. And when we checked in a CEC4 mutant, that's CEC4 is the mutant that releases heterochromatin from the periphery in early embryos, we found that the heterochromatin returned to the nuclear envelope as soon as you became an L1 larvae. So differentiation, there was a second pathway of heterochromatin anchoring induced in differentiated tissues. It was not K9 dependent, and it was not CEC4 dependent. So we did a screen to find what factors were involved in this sequestration in differentiated tissues. It was a limited screen because we had to score quantitatively the position of a heterochromatic array in each differentiated tissue, in each differentiated nucleus, so it wasn't just a de-repression screen. And there was a heroic effort. We had one hit. We always seem to only ever have one hit. But anyway, MRG1, it's a chromodomain protein, and it was the main hit. Now I'm going to skip a lot of data because I think the important thing is that you understand what MRG1 does. It works in parallel with CEC4, so they're both somewhat redundant. You have to knock out both of them to find a really strong background. So the K9 pathway is still functional in differentiated cells, but there's another pathway that compensates. And it's not only true for our array, but we did it genome-wide, and we found the same thing. This is just association of endogenous chromatin with the nuclear envelope. The MRG1 CEC4 double mutant in red here loses the association with the periphery. So what is MRG1? MRG1 is actually a chromodomain protein that binds euchromatin, and this is where the euchromatin comes in. It is called mortality-related gene 1. It's a chromodomain protein. It recognizes H3K36 di and tri. Its homologs in mammals and yeast are actually associated with histone acetyltransferases, TIP60, and certain deacetylases. It was totally counterintuitive. We had been studying sequestration. We studied something that released heterochromatin, and it was a euchromatic factor. We confirmed that it co-localizes with H3K36 methylation, and we got the same result by knocking out K36 methylation. We got the same release. So it was clearly a euchromatic mark, a euchromatic reader that was somehow talking to heterochromatin. So we tried to figure out what it was. MRG1, K36 methylation, they're clearly on euchromatin. They're not in heterochromatin. But when we knock out MRG1, we release heterochromatin in differentiated cells. You have to also knock out. So we asked whether it affects transcription. In fact, it did. But I'm just going to tell you, all the genes that got de-repressed were heterochromatic, had heterochromatic marks. So it was like euchromatic genes just didn't care about this euchromatic complex, but the heterochromatin did. So it acts indirectly. It does not act by turning down K9 or K27 methylation. We didn't get the same effect if we turned those down. So we knock out K9, K20 methylation, or K27 methylation, no effect. So it's not an indirect effect on other modifiers. So we decided that it's acting indirectly. It's sitting in euchromatin, binding something. When you take it away, maybe this something goes and attacks heterochromatin and then releases it. So this was our model. MRG1 is in euchromatin. If you knock it out, now HATs and HDACs or something is there to release heterochromatin. So we screened, and I'll just tell you the answer. We screened all HDACs and all HATs. And we asked, in an MRG1 mutant where the heterochromatin is now away, what restores it? And we got a super hit. It's CBP3, which actually is a homolog of CBP1, which is P300. It is one of the classic active marks, HATs. It's present on almost every active promoter. It's recruited by transcription factors to drive transcription. We also knocked out CBP1, which is an essential gene. That's why we actually CBP3 kind of downregulated it, but didn't knock it out completely, and we got the same effect. So CBP300 is a major K27 HAT. So our model was you knock out MRG1, CBP3 goes and invades. That would only be true if CBP1 was normally limiting in the cell. And so we overexpressed CBP1, and we had the same effect. Overexpression of CBP1 releases heterochromatin in a CEC4 mutant. How can this HAT just willy-nilly go and attack heterochromatin? Because you have them in the cell anyway. So we decided there must be a transcription factor there. And we screened all the genes that were upregulated for common transcription factors. And we enriched in the CEBP family transcription factor. It really came out strikingly enriched. A CEBP family motif. So we screened all the leucine zipper, BZIP factors for, again, restoring heterochromatin at the periphery. And again, we got just one hit that was significant. It was called ATF1. ATF1 is a CEBP homolog. These are BZIPs factors that bind usually in dimers, and they form different types of heterodimers and activate genes. So when you knock out ATF8, like knocking out CBP1, you restore the association of heterochromatin with the periphery in an MRG1 mutant. That means the MRG1 is really acting by releasing CBP and then allowing binding at the periphery. And sure enough, this transcription factor, ATF8, has binding sites in our heterochromatic reporter. So the model is this. This is the fruit of many years, actually. We know that in non-differentiated cells, CEC4 is a specific K9 reader and that monodi or trimethylation will sequester heterochromatin at the periphery. We know that in differentiated cells, this still works, but is redundant with another pathway that can be antagonized by knocking out MRG1. So there's redundancy in differentiated cells that is not found in embryos. So we know that we can overcome the MRG1 knockout by knocking out CBP1 or a transcription factor called the ATF8, DRB in mammals. And so that actually the euchromatic sequestration of a histone of a hat is as important as the CEC4 binding of the K9 for keeping heterochromatin at the nuclear envelope. What does this mean? And I think this is really important, so I excuse myself for running over. It means that it's not just that our genomes sequester away useless DNA at the nuclear envelope. It's that our genomes are packaged both in euchromatic domains and heterochromatic domains. And the euchromatic domains themselves sequester factors that are important for this segregation. And if you perturb the ability of euchromatin to keep its hats in euchromatic domains, or if you overexpress certain hats, you can destabilize the heterochromatin and allow it then to be released. So the organization of the genome is not what I always thought, the essential, only the realm of those heterochromatic marks being bound, but it's actually equally important. If you assume that CEC4 and MRG1 are equally important, that's what our data shows, it's equally important to keep euchromatic histone cell transferases away from heterochromatin. And what it also means is that pioneer factors, ATF8 and DRB is a pioneer factor. It can bind in heterochromatin. That pioneer factors will turn on genes in heterochromatin if the transactivating hats and mediator and so forth can have access. So it's very, very essential. And actually, if you go through the P300 literature, you find that there are many instances where misregulation of P300 is actually affecting genome organization, but more generally, the de-repression of tissue-specific genes in differentiated cells. So that's my message, that in differentiated cells, heterochromatin sequestration requires both an intact euchromatic compartment and anchors for heterochromatin. And these are my three heroes, Jan, Peter, and Daphne. Thank you.